So this video is going to be about Mendelian genetics. This is going to be part of a small new playlist I'm going to make about genetics. So starting off, Mendelian genetics are based off of three postulates, which are that unit factors exist in pairs, and that genetic characters are controlled by unit factors. So unit factors are basically literally just genes, right? Unit factors are another name for genes. The second postulate says that talks about dominance and recessiveness. In a pair of unit factors or a pair of genes, one unit is usually dominant and the other is recessive. And the third postulate talks about segregation and the concept of assorting independently. And it says that paired unit factors segregate independently during gamete formation. So this is just referring to the process of meiosis, and that meiosis is an important part of understanding Mendelian genetics and the variation in it. So moving on, some key terms that you should know are gene, which I already said, those are unit factors, or in this definition, the unit of inheritance. This is the thing that's going to help us actually transfer information from parents to offspring. Alleles are a specific version of a gene. So you're going to see this word a lot. You're going to see this word allele a lot when you're solving problems. So allele and genes are totally different. Alleles are found on the genes, right? Genes are the big thing, right? That's the main mode of inheritance. But alleles are different versions of those genes found directly on that unit of inheritance. So I gave an example right here where the gene is eye color, and alleles are actually the specific colors of the eye, right? So the gene that we're gonna be the gene the example I'm giving is eye color, and blue and brown are specific colors that are found in that gene, right? So the next term is genotype, and genotype is basically the exact versions of an allele, right? Or in other words, the genetic makeup of an individual. And then phenotype are the observable characteristics of a genotype. And then these two terms, homozygous and heterozygous, they're going to be, you need to understand these terms for like Punnet squares and pedigrees. So homozygous just means that you have two of the same versions of an allele, right? So for the example I gave right here is lowercase b, lowercase b. It could also be uppercase b, right? Now, for heterozygous, it's the definition is that two different alleles are found together, right? Two different alleles. And right here, we have a heterozygous, right? Individual, I guess. And it's supposed to be two different alleles. So big B, little b. So moving on now, these were just key terms and just basics of Mendelian genetics. Now we can move on to specific applications of Mendelian genetics, such as Punnet squares. So the first Punnet square are monohybrid crosses. Monohybrid crosses usually involve only one trait, so you have to keep that in mind. And in the word itself, it says mono. Mono in Greek means one, hybrid means a cross, right? So a monohybrid cross involves one trait. So in monohybrid crosses, there's still going to be the concept of dominance, where one allele is going to be dominant over another, right? And that other allele is usually going to be recessive. So let's do that. The best way to actually understand Punnet squares or any other applications is doing examples, right? So I gave an example right here where brown is dominant, blue is recessive, right? And once again, these are alleles, and the overall gene is the eye color. So let's say we cross two heterozygous individuals, right? Heterozygous meaning two different alleles, and so it's big B little b times big B little b. One of these individuals is the male or the father, and this other individual is the female or the mother. So when you're making a monohybrid cross, you want to make a table something like this, where you can relate two different actually four different alleles, right? Four different alleles creating four different offspring. So the first thing you want to do, so I'm going to put the male on top here and the female over here. So the male genotype is B, uppercase B, lowercase b. Female genotype is uppercase B, lowercase b. Alright? Now, when you're doing crosses, all you have to do is cross a male allele with a female allele, right? So, for this box, we're crossing this allele with this allele, and we get uppercase B, uppercase B. Then same idea, we cross this allele with this allele, then this allele with this allele, this one with this one. And when we do that, we cross all of those alleles, we get this. This is our completed monohybrid cross. So these alleles right here represent the parent alleles. These are the actual offspring that we get by crossing these alleles, right? So when looking at this monohybrid cross, we have a bunch of different offspring, right? Some have all capital letters, some have all lowercase, some have a mix of them, right? So when we're trying to name these alleles, these offspring, or like what they are, there's certain terms you have to use. So for this double uppercase offspring, we usually call this so BB is equal to, this is a homozygous dominant offspring, right? Homozygous meaning the same allele, and then dominant meaning it involves the dominant allele. And uppercase usually means dominant, and we have two of the same, which corresponds with homozygous. So uppercase B is a homozygous dominant offspring. So lowercase b would be a homozygous recessive. And once again, two of the same alleles, meaning homozygous, and lowercase alleles, right, equals recessive alleles. So now this last one now is a heterozygous offspring. So it's heterozygous. So it means that there's one of each allele, one uppercase allele, one lowercase allele, one dominant, one recessive. And usually when we're looking at something like this is a heterozygous dominant offspring, because the dominant allele always takes precedence over the recessive allele. So heterozygous. And there's nothing in Mendelian genetics that is called heterozygous recessive. The dominant allele will always take precedence over that recessive allele if they're found together. So in this case, when we're trying to actually determine the phenotype based off of these genotypes, we already know that brown equals to dominant, recessive, blue is equal to recessive. And we already found out what our offspring are based off of the monohybrid cross. So for brown color, right? We basically have right here 3 different offspring, right? And this heterozygous offspring is still brown since the dominant allele takes precedence, right? And this right here is 2 brown alleles, so obviously it's going to be a brown offspring, right? Brown colored offspring, brown eye color offspring. So the different offspring or different genotypes that can represent brown in this case are BB and BB. They both mean brown eyes, right? Now for blue, the only genotype we have here is lowercase b. That's the only genotype we have. If you want something to be recessive in a monohybrid cross, we need two of the same alleles, right? We need 2 of the same recessive alleles. So the genotype for blue is lowercase b, right? And based off these monohybrid crosses, we can actually find percentages of, like, say a question asks you, based off of this cross, what's the percentage? What's the probability of the offspring being blue-eyed? And in this, it would simply be 1 out of 4, right? 4 total offspring, and only 1 blue-eyed found in that cross. So 1 over 4. And for brown, we would say 3 out of 4, right? 3 out of 4, since there are 3 total brown offspring and over the total number of offspring, so 4, so 3 over 4. And this is just the simple way of understanding monohybrid crosses. There could be a lot more complicated ways to understanding this, but this is the basic idea. Now, Punnet squares don't necessarily have to show one trait is dominant over others. There are certain cases where either they could be a blending of traits, or there could be codominance, something like codominance, where 2 different dominant alleles are found in the offspring. So let's look at incomplete dominance first. So incomplete dominance is the blending. Involves blending of traits or alleles. So definition is when a dominant allele does not completely mask the effects of a recessive allele. Phenotypes show a blending of both alleles. So a perfect example for this is red and white flowers, or I think the specific name of the flowers are called snapdragons. So red and white flowers. When we're crossing, I'll use the name snapdragons. They are found in red colors and white colors, right? But when they're reproduced, so say we have a heterozygous snapdragons, and RR represents red colored snapdragons, and then WW represents white colored snapdragons. So these are how they're usually found, those flowers. But when we're trying to cross these 2 flowers, so let's say we start off with a cross between red and white, right? Red and white. We're gonna get offspring that have both alleles in there, both alleles in the offspring, but one isn't dominant over the other. So what happens is that the offspring have a color that is in between red and white, and that color is basically pink, right? So this RW is the color pink. This RW represents the color pink. And this is an example of incomplete dominance where there's going to be a blending of traits, since neither red or white are dominant over each other. So so this is the example for incomplete dominance. For codominance now, this is where both alleles in the genotype are seen in the phenotype, right? So there's going to be a type of sharing when we're crossing these alleles. So the example for codominance is the ABO blood type. In ABO blood types, A and B are usually the ones that are dominant, and O is the recessive blood type. So let's say we do a little cross right here. So like a little cross right here. So let's say we cross a person with AB blood type with a person with AO blood type, right? So that's basically AB blood type. So when we cross these blood types, we get AA, AO, AB, and BO. So what we see here are a lot of different offspring, but over here, in this example right here, AB blood type, you see both of these alleles, right? Both of these alleles, and once again, one is not dominant over the other, so both are going to be seen in the phenotype. So this is what you call AB blood type, right? This is what you call AB blood type, and A and B are both A and B are both seen on this in this blood type. But in this case right here, AO and BO, A is dominant over O, since O blood type is recessive, so this is overall A blood type, and B is dominant over O, and B is overall A is dominant. I mean B is dominant over O, and since O is the recessive blood type, this is overall a B blood type. And then over here we have AA, and these are just the same allele, so this is just the A blood type. But this is an example of codominance, where both alleles are found in the genotype. Now moving on, the test cross. The test cross is used to determine whether an organism with a dominant phenotype has a heterozygote, heterozygote, or a homozygote. This method is used by plant and animal breeders. So a test cross basically helps us determine which type of dominant was a certain parent based off of the offspring. So how do you do this? Or an organism with dominant phenotype is crossed with an organism that is homozygous recessive. So when we do this test cross to find basically the missing genotype of a parent, one result that can happen is that the F1 offspring are all heterozygous for the dominant allele. And the second result that can happen is that F1 offspring will be half heterozygotes and half recessive homozygotes. So there's going to be a 1-to-1 ratio in result 2. So let's actually try to draw out a monohybrid cross. Let's try to draw out a monohybrid cross with this. So let's say, so we know we're going to be crossing with a homozygous recessive. homozygous recessive organism, right? So let's say we have Y, and we're not gonna really say what Y is or what kind of gene it is. We're just gonna use this basic example. So we know it's going to be dominant, so one of the alleles has to be capital Y for the genotype of the missing parent, and we're trying to find what is it heterozygous or is it homozygous. So let's say the offspring are all heterozygous, right? This right here, since the offspring are all heterozygous, we already know that the missing allele of the genotype of the missing parent is obviously going to be lowercase y, right? When you cross a heterozygote with a homozygous recessive, we're going to get all heterozygote offspring. And this is exactly what happened here. So this is basically a basic example of this result 1. Now result 2 involves a 1-to-1 ratio. So we know we're going to be crossing with a homozygous recessive. We know one of the alleles, since we're trying to find a dominant parent, right? A dominant parent. So we're trying to find question mark, which so we already did all offspring of heterozygotes. Now say half of the offspring here are heterozygotes, right? Dominant heterozygotes, and half of the offspring are recessive, right? Recessive. So what happens here is that what happens here is that the missing allele based off of these offspring is lowercase y. So it's a heterozygote. So over here, I made a small mistake. This should be a capital Y. So over here, in this example, we are finding a homozygous dominant parent. Over here, we found a heterozygous dominant parent. And this is all based off of the offspring. Since we have half heterozygotes, half recessive offspring, it gives us a heterozygous parent. Over here, since we had all heterozygous offspring, we have a homozygous dominant parent. So this is how you just use the test cross. It's usually found in monohybrid crosses. So yeah. Now moving on, we have dihybrid crosses as well. So similar to monohybrid crosses involves one trait. Dihybrid crosses involves 2 traits. So it's a cross between 2 different genes that differ in 2 observed traits, right? So say one gene is brown. So in this example I gave, one gene is for eye color, one gene is for teeth size. So the alleles are brown eyes, blue eyes, big teeth, and small teeth, right? So it involves 2 different genes and 2 different alleles within those genes. So let's actually do this example right here. So this is a hybrid cross between two heterozygous parents, right? And they're both heterozygous for each gene. So the first thing we want to do in a dihybrid cross is determine the gametes, right? The gametes of each parent. So how you determine gametes is basically similar to what you might have learned in like algebra, like the distributive property. We can distribute these alleles to find gametes that have two alleles of each gene. So let's say we distribute this capital B. So let's distribute here and distribute here, right? we end up with the alleles B, T, and BT. Now we distribute again with the other blue-eyed allele, and we get the gametes BT and BT. And you would do the same thing with this parent as well if it was different, like a different genotype. But since it's the same genotype, we can just copy that right here as well. And we're going to be crossing this. So like this is a 16 box Punnet square, and it's going to have 16 total offspring. So just like a monohybrid cross, we're going to be crossing alleles, right? I mean crossing pairs of alleles in this case. So right here we're going to be crossing this with this, this with this, this with this, this with this, and so on, right? So let's complete this Punnet square. We get BBTT. We get BBTT. 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 So BBTT. Right here is BB. So this is the completed Punnet square, a dihybrid cross. So you see there's a bunch of different offspring. So let's actually circle the types of offspring, right? So starting from this corner right here, this genotype represents blue-eyed and small teeth offspring, right? Since blue eyes is recessive, lowercase t is represents small teeth. So if you have one of those offsprings, let's use a different color. Let's use orange. So right here we have blue eyes and big teeth right here, blue eyes and big teeth right here, blue eyes and big teeth right here. So this is blue eyes and big teeth. Now let's use a different color. Now we have brown eyes and small teeth right here, brown eyes and small teeth right here, brown eyes and small teeth right there. So we have 3 of those offspring. Now right here we have brown eyes and big teeth, brown eyes and big teeth, brown eyes and big teeth, this one too. So the rest of them are all brown eyes and big teeth. So we have 9 of those offspring. So overall, when we're crossing two heterozygotes for 2 different alleles, for 2 different genes, we get a 9:3:1 ratio, right, of different phenotypes and genotypes. So this is how you do a dihybrid cross, basically. And this example just represents a common example you'll see, which involves crossing 2 different heterozygotes. So moving on. So this is a small summary of all the monohybrid cross and dihybrid cross trends that you're usually going to see. So let's say if we cross, so starting off in monohybrid crosses, say if you cross a homozygous dominant with a homozygous recessive, homozygous dominant with a homozygous recessive, so that is basically homozygous dominant. Let's say it's capital B, lowercase b, lowercase b. So when we're crossing that, the F1 generation, right? The first time we cross both of these parents, all of them, all the offspring are going to be heterozygous dominant. So they're going to be B, 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 right? Going to be BB. But when you, let's say in the F2 generation, which means that we're going to self-cross one of the F1 offspring. So since this was the F1 offspring, we're going to self-cross this with itself. So we're going to cross with another BB, heterozygous BB. So when we cross two heterozygous in a monohybrid cross, we get three dominant offspring and one recessive offspring. And these three dominant offspring are composed of basically two heterozygous offspring and one homozygous, right? So heterozygous is basically BB, homozygous BB. But they still code for the same dominant allele, right, in the phenotype. So overall, this is whenever you have two heterozygous parents reproducing in a monohybrid cross, that's going to be a 3-to-1 ratio. But if you look at the dihybrid cross, if you cross a homozygous dominant with a homozygous recessive, in the F1 generation, all of the offspring are going to be dihybrid, right? Are going to be dihybrid. Now, if you self-cross a dihybrid from the F1 generation to make the F2 generation, we're going to get what we noticed here previously in the dihybrid cross example, where we crossed two dihybrids, right? These are dihybrids. They're heterozygous for two different genes. And what we noticed was that we get a 9:3:1 ratio, right? 9 of the dominant allele, dominant offspring for both genes. Then we have 3 offspring that are dominant for one gene, but recessive for the other. Then we have another 3 offspring that are dominant for one gene and recessive for the other. And then we have only one double recessive offspring. So this is just a small summary of the general trends you're going to see when you're doing dihybrid crosses and monohybrid crosses. And it's recommended to just memorize these ratios, so you don't have to do a whole dihybrid cross or a whole monohybrid cross to actually find certain offspring, since you already know the ratio, so you'll know the actual genotypes as well. So moving on now, we have the topic of independent assortment and linkage. So in linkage, two different genes are inherited as a unit, right? As a unit, due to their proximity to each other on a chromosome. So, based off this definition, let's just do a small example first to understand how linkage works. So let's say we're crossing two purebred pea plants, right? With the genotypes, the capital Y, capital R times lowercase y, lowercase y, lowercase r, lowercase r. So first let's just point out which alleles are dominant, which alleles are recessive. The dominant alleles are Y and R. Recessive alleles are lowercase y, lowercase r. So parent 1 So let's say this is parent 1, this is parent 2. So parent 1 has a genotype of capital Y, capital R, capital R. And since these are two different genes, involves two different traits, two different genes, first we want to determine what are the gametes of each parent, right? Just like how we did it in dihybrid crosses, where we distributed one allele to each of the other alleles. We do the same thing here for each parent. So in this first parent, we're gonna be distributing this Y to this R, this Y to this R, but they're all gonna end up with the same gamete. So we can just write the first gamete we get is going to be capital Y, capital R, right? Now the second parent has a genotype of lowercase y, lowercase r, lowercase r. And we do the same thing, and we get a gamete of lowercase y, lowercase r, right? So what these two gametes represent right here are linked gametes, right? We're gonna say these are linked gametes. So when we have linked gametes and we try to actually cross the parents, so in these parents, these gametes are linked. So when we perform a dihybrid cross, what we're gonna get for the F1 offspring, let me write F1. When we perform our actual dihybrid cross, I'm not gonna draw that whole dihybrid cross out, since this is a very small cross. So when we actually do this cross, it's going to be something like, I'm just gonna make a little monohybrid cross. So it's going to be YR. So this is the parent 1. Now parent 2 is this right here. And when we cross this, we get these offspring for the F1. And you see they're all the same offspring, right? They're all the same offspring, same genotype. So we can just say that the genotype we observe is Y, capital R, lowercase y, lowercase r. And in these offspring, both of these alleles are linked, right? This capital Y is linked to this capital R, lowercase y is linked to this lowercase r. So they're linked alleles, right? Or in this case, linked genes. So when we actually cross this right here, let's say if we self-cross this to find the F2 offspring. So let's say if we self-cross this, but remember, we're not gonna forget about the linked portion, right? The linked concept, linked alleles. We have to remember that these are linked alleles, right? Y, capital Y is linked to capital R, lowercase y is linked to lowercase r. So if we perform a self-cross, so let's say if we perform a self-cross to get the F2 offspring, we have to remember, we have to keep the linked alleles together. So we're going to be crossing YR, and YR, right? When we, just like a monohybrid cross, we cross this with this, we get YR. We cross this with this, you get YR, and so on. And we get these offspring right here, right? As you can see, there's still a 3-to-1 ratio, right? There's still a 3-to-1 ratio here. However, there's still a 3-to-1 ratio. However, there's there are different pairs of alleles. So like if we rearrange how we write this, I wrote it like this so you can just see, you guys can just see that these alleles are actually linked. But if we write it where we pair up the same alleles or the like alleles, so let's say, so I'm looking at this one right here first. So let's say if you rewrite this as WWYRR, then we rewrite YYRR, and then YYRR. Oh, there's another one of these too. So you see, there's still, like, you could still say that this right here means yellow and round, right? See right here, capital Y means yellow, capital R means round, lowercase y means green, lowercase r means wrinkled. So we still have a 3-to-1 ratio right here, where we have yellow and round, yellow and round, yellow and round, and then one offspring is green and wrinkled, 3-to-1 ratio. So we can still say still a 3-to-1 ratio, but the gametes are linked, right? 3-to-1 ratio. Linked gametes. So moving on. Oh, I guess. So this is really it for the basics of Mendelian genetics. I'm gonna be posting more videos as well, going over some more advanced topics and actual problems in Mendelian genetics. But for now, this is basically the basics.